So good morning. Now it's my turn and I'm going to talk about the alveolata representatives. Okay. Here we have again our eukaryotic tree of life and we are here. Okay. And what do we find in the alveolates? We have this group of apicomplexa, okay, that is mainly composed by parasites. As Marco said before, we can find here the agents causing malaria, toxoplasmosis, etc. So it's an important parasitic group, but not a zoosporic one. Then we have here ciliates and dinoflagellates, important components in the planktonic environments. And then we find here two small groups composed exclusively of parasitic organisms. And inside those groups that are quite heterogeneous in terms of host and so on, we have some that infect algae. So I'm going to focus on those. I'm going to give an overview of the diversity, the known diversity inside these two groups, and I will focus only on those infecting algae. Compared to the other groups, mainly the chytrids, we only know a few species in there, so we can go species by species and genus by genus because we don't know much in that. First, I will begin talking about Perkinsians or Perkinsozoa, depending on the taxonomic rank that we are talking about. We can find these two names. And the first species described was Perkinsus, that infects clams, or I cannot see the text here. And well, it infects, we can find them in the tissue of these clams. And here we have the life cycle. As we know, we are talking about zoosporic parasites. So we have here the motile stage, the zoospore, that penetrates the host via the filtration, the active filtration of the shellfish. And then it produces these trophocytes that are the vegetative growing stage in the host. And at some point, they produce the sporangium that begins dividing, producing the zoospores that will be released through these structures, these small tubes, okay? Then we also have another group called the Exalidae that produce these tumor-like formations in fish. I even said that Perkinsus is found in marine environments, this one again in marine environments. They have this strange name because these infections were known for a long time, but didn't know the causative species, and they call them X cells. At the end, they found the molecular information, they could describe them, and then they call them Exalia, Garixalia, Salmoncellia. Okay. And then that one is from freshwater. We have this clade here that is composed mainly by molecular environmental sequences, but they could relate those sequences with an infectious agent in tadpoles, okay? So we can find these parasites in freshwater environment infecting the tadpoles. And then let's move to this group, Parvilociferaceae, that are those that infect the algae. The first two species described was Parvilocifera infectans at the end of the last century in the North Sea. And in this case, again, they are marine and they infect dinoflagellates. We have the life cycle again here. It's really similar to the life cycle of all the previous organisms that we have seen so far. We have the motile zoospores that penetrate the host, the dinoflagellate. They form the trophocyte. And this trophon begins feeding or consuming the cell of the dinoflagellate, grows in size, and once it has completely consumed the host cell, then it transforms into the sporon, the sporocyte, that we don't have the host cell anymore, it's consumed, so we can consider this a free-living stage. And then it begins dividing to produce the new zoospores that will be released to the environment. And here we can see the development of this cellular division, the nuclear divisions to form the new zoospores. Then we have different genera described here with different species. The Parvilocifera ones produce this round sporangia, and the zoospores are released through holes in the surface. Okay, all the different species are really similar between them. And depending on the shape and the size and the number of the holes, we can discriminate them. This one is called multicavata because it has a lot of small holes. This one is called catillosa because it has this protuberant and big holes. And then we can find other genera that don't have holes, but they have processes here. We can see in the surface these processes. And then we have other genera that don't have holes, but small structures to release the zoospores. In this case, Tuberlatum has short tubes and the cyst wall is smooth. And then we have two other genera here that produce tubes and they are smooth again. Besides these structures of the sporangia, we can also discriminate the different genera and species by the size and the shape of the zoospores. In this case, the zoospores have two flagella, one longitudinal one and one transverse or anterior one. We can see it here. And for Parvilocifera, the zoospore is elongated. And for the other two genera or three genera, they are sigmoid. They have this, I say, tack shape, okay? And the swimming behavior is different. So this is how we can discriminate the different components in this family. So these are the different species described, the different images here. We can see the trophon inside the cell growing. You can see it here too. At some point, the host is not there anymore, it's consumed, and then they begin dividing to produce the zoospores. Then the zoospores are released. We have this empty sporangia, and here we can see the wall, the hole from where the zoospores escape. You can see it here, not so clear here. And then I have a video, I don't know if it will work now. Okay. So this is the material sporangium, okay? This is stage here, this is stage here. Here we have a hole, and we see the zoospores that are released through this hole. Okay, at the beginning, they are swimming slowly, and at some point they activate and they swim really fast and they escape, trying to find another host to infect. You can see that there are hundreds of zoospores inside the sporangium, and it depends on the size of the sporangium. And it also depends on the size of the host. So the bigger the host, the bigger the sporangium. And when you have multiple infections in the same host, then they have to share the results, okay? And then the sporangia are smaller. So it depends on the food they have, they can grow more or less. Then there is the next genus, Tuberlatum, and as I said before, they don't have holes, but they have these small tubes, or it's like gems, that at the end they open. You see here that they open, and then we have the zoospores here that the shape is different. They have this sigmoid shape, okay? And the same happened for this one, Dinoborax. In this case, they produce this tube, okay? When they stop feeding, the trophon forms this tube, and then it begins the division to produce the zoospores. And at the end, the zoospores are released through this tube. You can see here, the tube is opened, releasing this is the shade, the theca of the dinoflagellate. So it's inside the dinoflagellate, the tube emerges outside, and then the zoospores are released. And here you can see the shape of these zoospores. And again, stop. Let's see. I think I have to do it. So this first one is a time lapse video. Okay, and here we have the infection. This is the dinoflagellate cell. It is consuming the host. At some point, it will produce the tube. Just to see a little bit how it looks like when you follow the infection in a long time. The video then stops here, but it's beginning to produce the zoospores now. And then in the other one, we can see the release of the zoospores. A little bit dark, but have the tube here, and then the zoospores are released here. And you can see that the swimming behavior is different than for the Parvilocifera one. The Parvilocifera were really swimming really fast. In this case, they are not so fast. The zoospores are bigger and larger, okay? And again. all the zoospores go around. So another one that is very similar to Dinoborax. And then last year, 2 new genera were described that they do not match with the features of the previous ones. Okay, in this case, we have this Maranthos that it forms this external wall, and then the zoospores are displayed in this radial disposition around the cell. And then when they are ready, they detach, the cell wall breaks, and then the zoospores are released through this external cell wall. And here we can see again the shape, the sigmoid shape of the zoospore. Let's see the video. Yeah, it moves. So the zoospores are similar to the previous ones, but they are not so well organized than before, going one and the other through the folds, but being immersed in this unorganized way. Okay. And the other one is this Pararosarium, that is even more different than the others because at some moment of the life cycle, the parasite emerges from the wall, okay? And then it produces these divisions extracellularly, that is called palintomy, producing these long chains that the size of the different cells is being reduced, forming these smaller ones until they are individualized as a zoospore. Okay, so this is clearly different to the previous ones, but it also belongs to this Parvilluciferaceae family. So, summarizing a little bit the information that is known for this group, they only infect dinoflagellates. They are exclusively marine and commonly found in coastal environments. There's scarce information about the distribution and occurrence of all these species. Likely, they are globally widespread when you look for the molecular signal of some of them, but we don't really know. And regarding the specificity, they can be considered as an intermediate specific or a generalist inside dinoflagellates. So they are able to infect many different species within dinoflagellates, not all of them, or maybe some orders, depending on some genera. But we also find differences regarding strain specificity, okay? The same strain of a parasite can infect one strain of a host of the same species, but not the other. So there's a strain specificity, and also happens the same for the parasite. So we have a matrix of infections and different levels of prevalence and infectivity. We don't really understand why, what makes that you can infect one strain and not the other of the same species. But well, so they can be considered as generalists, but okay. And for sure, they have host preferences. These are results from metabarcoding, but here we sampled different locations from the Catalan coast in the Mediterranean during blooms of different dinoflagellate species that is represented by these different colors. And we find that depending on the host producing the bloom, we find one parasite on another belonging to this group. Even though all of them could be able to infect all of them, all the different dinoflagellates, they have host preferences. They are competing between them, and then the one that is able to infect more efficiently the host species is the one that is winning in the environment at that moment, okay? And how do they infect? They are density dependent, and they infect on actively growing host cells. This is data from field sampling. And in black, we have the abundances of the host, the Alexandrium minutum. And we see that once it reaches threshold abundance between 10 to 4, 10 to 5 cells per liter, this is the moment when we find the infections in the environment. That is represented by these vertical shadowed areas, okay? So they need, or at least we need, that the host is at that abundances to be able to detect these infections in the environment. They must be there somewhere when they don't have the host, but we don't really know where. The sediment, infecting other secondary hosts at low abundances, we don't know. But at the moment they have this threshold of abundance of the preferred host, then have the explosion of infections in the field. So in sum, these are the different groups I showed you. They are really nicely grouped by phylogeny, except those two last, Pararosarium and the other one that they not really fit within these dinoflagellate parasites. Most of them are marine, except those from tadpoles, but we have a lot of molecular information. And actually, in freshwater environments, Perkinsea are a component that is usually found in those kind of sequencing data. But we have no idea about who they are, except those that infect tadpoles. We don't know who are they. So there is something to be known in freshwater environments about this Perkinsea. And now I'm going to, again, I'm going to change the group. I'm going to talk about the Sindiniales. I don't know what happened. Maybe it's presentation. So, in this case, we have different molecular groups that they were called 1, 2, 3, 4, 5. We can find this group under the name Sindiniales or MALPs, that means marine alveolates. And for 3 and 5, we don't know, we only have molecular information, we don't know who they are. And I'm going to talk a little bit about 1, 2, and 4. Again, they are exclusively parasites. We can find different kinds of hosts, from fish, crustaceans, algae, protists, etc. Only marine representatives are known for that group. They have, again, biflagellate zoospores, that in this case they are called dinospores. And sometimes they see that they have different size and different shapes. And they have related that with the sexual reproduction. So one individual can produce one kind of dinospore, another individual can produce another kind, and maybe they are doing a sexual reproduction in that case. And the trophons conduct nuclear divisions, producing a multinucleate cell or a plasmodium. Inside group 1, we have this Eudobosquela that, as you can see, this produces this palintomy, as we saw before for the other Pararosarium. It infects ciliates, the tintinids. Here we have the tintinid that live inside this cup. This is the ciliate, and here is the parasite, okay? And it begins dividing and feeding on the host and producing these long chains that at the end, they will produce the spores. We also have this Ichthyodinium that parasites, that is a parasite of fish, of eggs of fish. Anyway. And then in group 2, we find Amebopria, that is also important because it infects dinoflagellates again. It's an endoparasite, as for the Perkinsids. We have here the trophon that feeds on the host, grows in size. And then we have a special stage here that is called the beehive, trophon stage. I don't know if you can see, it has some lines here, which resembles the beehive. And then it forms this vermiform stage that emerges from the host cell and it swims. Okay, so at this stage, you have like this worm that goes away, swimming, and then it at the end it produces the different zoospores of the Amebopria. And well, in this case, it seems that they are highly specific, these parasites, and they are quite abundant in marine oceanic waters, or at least in molecular surveys. Actually, here is the molecular information, and we see that depending on the host that they are infecting, they have different molecular information for all of them. So it seems that it's really specific, even also for the molecular sequences they have. So maybe they are different species at the end, but this is what we know so far. And for group 4, we have Syndinium, that is the one that gave name to the group, that produces this multicellular plasmodium that will infect the copepods and radiolarians. You can see here inside the body, this long thing here. This is the plasmodium with multiple cells inside. And again, this is a marine representative. And finally, we have another Apodinium, but in this case, they are parasites of crustaceans, and they produce these arachnoid trophones in the tissue of the crabs and lobsters and so on. And finally, we have these two guys here. Actually, they do not cluster inside the mouths or the Parkinson's, but in between, it's not clear the relationships with the other ones, and in fact, they are ectoparasites, in contrast to the others that all of them were endoparasites, okay? So they infect copepods again, they are marine again, and you can see that they form these sacs here that are attached to the copepod, and then they follow their life cycle here. And that's it.